All around us, there is a world we cannot see. It is a world teeming with life, a bustling, vibrant community of the very small. These tiny living things are called bacteria. They are everywhere. They drift in the air we breathe. They float in the water we drink. They live deep within the soil beneath our feet. Even on our own skin and inside our bodies, countless millions of these microscopic beings make their home. They are the oldest form of life on our planet, the silent architects of the world as we know it today. And though they are invisible to our eyes, their influence is truly immense. These creatures are almost unimaginably small. A single grain of sand could hold millions of them. For most of human history, we did not even know they existed. It was only with the invention of the microscope that we first peered into this hidden kingdom. What we discovered was astonishing. It was a revelation that changed our understanding of life itself. We learned that we are not alone, not even in our own bodies. We are part of a vast, interconnected web of life, and bacteria are the foundational threads that hold it all together. Their story is the story of life on Earth. Imagine a world without them. It is simply not possible. They are essential workers, performing jobs that no other living thing can. They break down dead matter, recycling vital nutrients back into the ecosystem. They help plants grow, protect our bodies from harm, and even help us digest our food. They are the great chemists of the natural world, transforming elements and creating the very conditions that allow larger, more complex life like us to exist. This microscopic realm is not a distant alien place. It is deeply, intimately connected to our own existence in every possible way. So, let us embark on a journey into the world of the small. We will explore these remarkable organisms, these microscopic marvels. We will learn what they are, how they live, and the profound impact they have on our planet and on our lives. It is a journey that reveals the extraordinary complexity and beauty that can be found in the simplest of life forms. The world of bacteria is a world of wonder, waiting to be discovered. It is a story of survival, adaptation, and the enduring power of life itself. What then is a bacterium? At its heart, it is a single cell. That is all. It is a complete, self-contained living being, all packed into one microscopic package. Unlike the cells that make up our own bodies or the cells of plants and animals, the bacterial cell is wonderfully simple. It belongs to a group of organisms called prokaryotes. This name tells us something very important about its structure. Pro means before, and karyon means nucleus. A prokaryote is a cell that came before the nucleus. It has no central command center, no membrane-bound core holding its genetic information. It is life in its most fundamental form. Instead of a nucleus, a bacterium keeps its genetic blueprint, its DNA, coiled up in a region of the cell called the nucleoid. This single circular chromosome contains all the instructions the bacterium needs to live, grow, and reproduce. Floating around it in the jelly-like cytoplasm are tiny structures called ribosomes, which are responsible for building proteins. Everything is enclosed by a cell membrane, which acts like a gatekeeper, controlling what comes in and what goes out. Surrounding this membrane is a tough, protective cell wall. This wall gives the bacterium its shape and protects it from the outside world. It is an elegant and efficient design. This simple structure is one of the keys to the success of bacteria. Without the complexity of a nucleus and other internal compartments, they can operate with incredible speed and efficiency. They can absorb nutrients and expel waste very quickly. They can copy their DNA and divide into 2 new cells in a matter of minutes. This allows them to adapt rapidly to changing environments, a skill that has allowed them to colonize every corner of our planet. Their simplicity is not a weakness, it is their greatest strength, a masterpiece of evolutionary engineering that has stood the test of billions of years. And so, this tiny, single-celled organism is a marvel of self-sufficiency. It carries its own instructions, builds its own parts, and powers its own existence. It is a testament to the fact that life does not need to be large or complex to be successful. In the world of bacteria, we see the very essence of what it means to be alive. It is a blueprint for life that has been so successful, so robust, that it has persisted for ages, long before the first dinosaurs roamed the earth, and will likely persist long after we are gone. If you could shrink down and wander through the microbial world, you would find yourself in a gallery of varied and beautiful forms. Bacteria are not all identical blobs. They come in a wonderful variety of shapes. These shapes are not random. They are a fundamental part of a bacterium's identity, helping scientists to classify and understand them. There are 3 main shapes that we see most often. Each one is perfectly suited to the bacterium's particular way of life. It is form and function, working in perfect harmony, even at this microscopic scale. The first and simplest shape is the sphere. These spherical bacteria are called kochi, from the Greek word for berry. They can exist as single independent cells, like tiny marbles rolling through their environment. Sometimes they stick together after they divide. If they form a pair, they are called diplokochi. If they form a long, beautiful chain, like a string of pearls, they are known as streptokochi. And if they cluster together in a formation that looks much like a bunch of grapes, we call them staphylocochi. Each arrangement tells a story about how they grow and interact. Then there are the rod-shaped bacteria. These are called bacilli, from a Latin word meaning little stick. Bacilli are incredibly common and can be found in a vast range of environments, from the soil to our own intestines. Like the kochi, they can be solitary or they can remain attached after dividing, forming pairs or long chains. Some bacilli have an additional feature. They can form a tough, dormant structure called an endospore. This allows them to survive in harsh conditions, like extreme heat or drought, waiting patiently until conditions improve. It is a remarkable survival strategy. Finally, we have the spiral-shaped bacteria. These are perhaps the most elegant of all. They are known collectively as spirilla. Some are gently curved, like a comma, and are called vibrios. Others are shaped like a corkscrew, with a rigid body that twists as it moves. A third group, the spirochetes, are long, thin, and flexible, and they move by wriggling their entire body in a mesmerizing snake-like motion. This spiral shape is particularly useful for moving through thick, viscous fluids, like mud or mucus, allowing them to burrow their way to where they need to go. Bacteria are the true pioneers of our planet. They are ancient life. The first bacteria appeared on Earth over 3.5 billion years ago, when the planet was a very different place. The air had no oxygen, and the world was hostile and volcanic. Yet in this primordial soup, life began, and it began with bacteria. For billions of years, they were the only inhabitants of Earth. They slowly and patiently transformed the planet, producing the oxygen that would eventually fill the atmosphere and make it possible for other, more complex forms of life to evolve. We owe our very breath to these ancient microbes. Their incredible age has given them time to adapt to every conceivable environment on Earth. There is almost no place where bacteria cannot be found. They are truly cosmopolitan. They float high in the atmosphere and have been discovered deep within the Earth's crust, miles below the surface. They thrive in the freezing ice of the Antarctic and in the boiling water of hot springs and deep-sea hydrothermal vents. They live in water that is saltier than the sea and in pools that are as acidic as vinegar. Their resilience is nothing short of breathtaking. They are also our constant companions. Our bodies are not just our own, they are ecosystems, home to trillions of bacteria. This community of microbes is known as our microbiome. They live on our skin, in our mouths, and most famously, in our gut. The number of bacterial cells in our body is thought to be roughly equal to the number of our own human cells. This is not an invasion. It is a partnership, a symbiotic relationship that has evolved over millions of years. These resident bacteria help us in countless ways, forming a crucial part of what keeps us healthy. This ability to live everywhere is due to their astonishing metabolic diversity. While we humans can only get energy from eating organic matter and breathing oxygen, different types of bacteria have evolved a huge range of chemical tricks to survive. Some get energy from sunlight, just like plants. Others can eat things that would be toxic to us, like iron, sulfur, or even hydrogen gas. This chemical flexibility is what allows them to make a home in places that we would consider completely uninhabitable. They are the ultimate survivors, masters of adaptation in a changing world. The life of a bacterium is often very short, but their ability to reproduce is, honestly, just extraordinary. When conditions are right, with plenty of food and a comfortable temperature, bacteria can multiply at a dizzying speed. They do not reproduce in the same way as animals or plants. They do not need a partner. Instead, a single bacterial cell can create a whole population all by itself. This simple yet incredibly effective method of reproduction is called binary fission. The name says it all. Binary means two, and fission means to split. The bacterium simply splits into two. It is a process of perfect cloning. The process begins when the bacterium grows to a certain size. First, it must copy its genetic information. The single circular chromosome of DNA makes an exact duplicate of itself. The cell now has two identical sets of instructions. These two chromosomes then move to opposite ends of the elongating cell. The cell wall and cell membrane begin to pinch inwards in the middle, creating a partition between the two chromosomes. This partition, called a septum, grows until it completely divides the original cell into two separate smaller cells. Each of these new cells, called daughter cells, is a perfect genetic copy of the parent cell. They are clones, and they are ready to start the process all over again. Under ideal conditions, this can happen very quickly. Some bacteria, like the common E. coli, can divide every 20 minutes. This means that one single bacterium could, in theory, produce billions of descendants in less than a day. This rapid exponential growth is what allows a small number of bacteria to quickly form a large colony, or in the case of an infection, to overwhelm a host's defenses. This method of reproduction is the key to their evolutionary success. While binary fission produces identical clones, bacteria have other clever ways to introduce genetic variation. They can exchange small pieces of DNA with each other in a process called conjugation. They can also pick up stray DNA from their environment. This ability to share genes allows for new traits, such as antibiotic resistance, to spread rapidly through a population. It is this combination of rapid cloning and genetic sharing that makes bacteria such formidable and adaptable organisms, constantly evolving to meet new challenges. In the vast world of bacteria, we find both our greatest allies and some of our most dangerous adversaries. The overwhelming majority of bacteria are not harmful to us. In fact, many are absolutely essential for our health and the health of the planet. These are the helpful bacteria, our microscopic friends. The bacteria living in our digestive system, for example, are vital. They help us to break down food that our own bodies cannot digest, and they produce essential vitamins, like vitamin K and some B vitamins, that we need to stay healthy. They are our partners in digestion. Beyond our bodies, helpful bacteria are the tireless recyclers of the natural world. When plants and animals die, bacteria and fungi get to work, decomposing the organic matter. This process breaks down complex materials into simple nutrients, returning them to the soil so that new plants can grow. Without these decomposers, the world would be buried under a mountain of dead material, and the cycle of life would grind to a halt. They also play a crucial role in agriculture, with some bacteria living on the roots of plants and fixing nitrogen from the air into a form the plants can use as fertilizer. We have even harnessed the power of bacteria for our own purposes, particularly in the production of food. The tangy flavor of yogurt, the sharpness of cheese, and the sourness of sourdough bread are all thanks to the work of specific types of bacteria. They ferment sugars in milk and flour, producing acids and other compounds that preserve the food and give it its characteristic taste and texture. This partnership between humans and microbes is ancient, a form of biotechnology that we have been using for thousands of years to create delicious and nutritious foods. However, a small minority of bacteria are pathogenic, meaning they can cause disease. These are the harmful bacteria, the germs that we rightly fear. Bacteria like Streptococcus pyogenes can cause strep throat, while Salmonella can cause severe food poisoning. These bacteria cause illness by invading our tissues or producing powerful toxins that disrupt our body's normal functions. For centuries, bacterial infections were a leading cause of death. Fortunately, we have a powerful weapon against them: antibiotics. These are medicines that can kill bacteria or stop them from multiplying, and their discovery revolutionized modern medicine. To study a world that is invisible to the naked eye, scientists need special tools. The most fundamental of these is the microscope. The invention of the microscope in the 17th century opened up the microbial world for the first time, allowing pioneers like Antony van Leeuwenhoek to see what he called animalcules swimming in a drop of water. Modern light microscopes are far more powerful, able to magnify objects thousands of times, making individual bacterial cells clearly visible. Through the eyepiece of a microscope, a hidden universe of shapes and movements is revealed. However, seeing bacteria is only the first step. Most bacterial cells are nearly transparent, making them difficult to see clearly, even with a powerful microscope. To solve this problem, scientists use stains. Stains are special dyes that stick to bacterial cells, giving them color and making them stand out against the bright background. Different stains can reveal different things about a bacterium's structure. One of the most important staining techniques in microbiology is called the Gram stain, developed by Hans Christian Gram in the 19th century. It is a cornerstone of bacterial identification. The Gram stain is a clever procedure that separates almost all bacteria into two major groups: Gram-positive and Gram-negative. The difference lies in the structure of the bacterial cell wall. Gram-positive bacteria have a thick cell wall that traps a purple crystal violet dye, so they appear purple under the microscope. Gram-negative bacteria have a much thinner cell wall, which does not hold the purple dye. They are then counterstained with a pink dye, safranin, and so they appear pink or red. This simple test gives scientists vital information about a bacterium, helping them to identify it and even predict which antibiotics might be effective against it. Besides their shape and Gram-stain reaction, scientists use a whole battery of tests to learn about a bacterium's identity and capabilities. They might test what a bacterium can eat by growing it on different food sources. They can test whether it needs oxygen to survive. Bacteria that require oxygen are called aerobes, while those that can live without it are called anaerobes. Some, called facultative anaerobes, are flexible and can live with or without oxygen. Each test provides another clue, helping scientists to build a complete profile of the microbe they are studying, like a detective solving a microscopic mystery. To study bacteria properly, scientists need more than just a few cells on a microscope slide. They need a large population to work with. The process of growing bacteria in the laboratory is called culturing. The basic idea is simple. You provide the bacteria with everything they need to thrive, and then let them multiply. The food that scientists prepare for bacteria is called a culture medium, or media. This is like a nutrient broth or a jelly, packed with all the proteins, sugars, minerals, and water that the bacteria require for growth. Culture media can be liquid, like a broth in a test tube, or it can be made into a solid gel using a substance called agar. Agar is a jelly-like material derived from seaweed. It is perfect for growing bacteria because it creates a solid surface, but the bacteria themselves cannot eat it. The nutrient-rich agar is typically poured into a flat-covered dish called a petri dish. A scientist can then introduce a tiny, invisible sample of bacteria onto the surface of the agar. The dish is placed in an incubator, a special oven that keeps it at the perfect temperature for the bacteria to grow. As the bacteria multiply through binary fission, they do not spread out all over the dish. Instead, they stay put, and a single bacterium will divide over and over again, creating millions and millions of descendants all piled up in one spot. This visible clump of bacteria, which started from a single cell, is called a colony. Each colony is a pure culture of millions of identical clones. Colonies can have different shapes, sizes, textures, and even colors. An experienced microbiologist can often get a good idea of what type of bacterium is present just by looking at the appearance of its colonies. This technique of growing bacteria on an agar plate is fundamental to microbiology. It allows scientists to isolate a single type of bacterium from a mixed sample, like soil or water. By picking a single isolated colony and transferring it to a new plate, they can create a pure culture, which is essential for accurate identification and testing. Culturing is the first step in diagnosing an infectious disease, in testing for antibiotic susceptibility, and in countless research applications. It is the art of making the invisible visible, transforming a few single cells into a thriving, observable population. The study of bacteria, the field we call microbiology, is more than just an academic curiosity. It is a field of immense importance that touches almost every aspect of our modern lives. Understanding these microscopic organisms helps us to protect our health, feed our population, and safeguard the future of our planet. Every discovery we make in the microbial world has the potential to bring about profound changes for the better. The journey into this unseen world is a journey towards a healthier and more sustainable future for all of humanity. In medicine, the importance of studying bacteria cannot be overstated. By understanding how harmful bacteria cause disease, we can develop new ways to fight them. This includes creating vaccines to prevent infections and discovering new antibiotics to treat them. As some bacteria become resistant to our current medicines, the search for new ones becomes ever more urgent. Studying the helpful bacteria of our microbiome is also opening up new frontiers in health, revealing how these microbial partners influence everything from our digestion and immunity to our mental well-being. In industry and environmental science, bacteria are powerful tools. We use them to produce medicines like insulin and industrial chemicals. We use them in bioremediation, a process where bacteria are used to clean up pollution, such as oil spills or toxic waste, by breaking the harmful chemicals down into harmless substances. They are essential in waste treatment plants, where they purify our water. As we face global challenges like climate change, scientists are exploring how bacteria can be used to create biofuels or capture carbon from the atmosphere. The study of bacteria reminds us of the profound interconnectedness of all life on Earth. It teaches us that even the smallest organisms play a crucial role in the grand scheme of things. It shows us that life is resilient, adaptable, and endlessly inventive. There is still so much we do not know. It is estimated that we have identified only a tiny fraction of all the bacterial species that exist on our planet. An entire world of discovery still awaits us, a world of new medicines, new technologies, and a deeper understanding of life itself, all hidden within the simple, elegant form of a single bacterial cell.